Hello everyone, welcome back. In today's episode, we will discuss about error correction mechanisms adopted by aminoacyl tRNA synthetases during charging of tRNAs. Imagine if your cells made mistakes every time they built a protein. Life wouldn't function the way it does. So, how does the cell ensure the near perfect accuracy? The answer lies in a remarkable proofreading system carried out by aminoacyl tRNA synthetases, the very enzyme that joins one amino acid to its cognate tRNA during charging of tRNA. Aminoacyl tRNA synthetases have a difficult job. Each enzyme distinguishes one out of 20 different amino acids, and they must differentiate cognate tRNAs from total set of perhaps 100 in all. And while doing so, it must add correct amino acid to its cognate tRNA. This job is really difficult because there are so many amino acids which are near similar to each other. They just differ from each other by just one hydrogen atom or one simple small group. So how this near perfect accuracy is achieved? To explain this, there are two theories. The first one is called the kinetic theory of proofreading. In this video, we will concentrate on the kinetic theory of proofreading done by tRNA, aminoacyl tRNA synthetases. Let us go back to charging of tRNA first. So we all know that charging occurs in two steps. In the first step, an amino acid is activated using ATP, and in this process, aminoacyl AMP is formed with the release of the pyrophosphate. This pyrophosphate is eventually lysed by pyrophosphatase, releasing large amount of extra energy which pulls this reaction in the forward direction. In step 2, the aminoacyl AMP formed during the first step is then reacts with the cognate aminoacyl tRNA, forming aminoacyl tRNA and releasing AMP as a byproduct. This happens because this aminoacyl AMP, in presence of tRNA, amino acid has more affinity for the tRNA compared to AMP. So it gets esterified to the tRNA, releasing AMP as a byproduct on the active site of the enzyme. The enzyme that carries this reaction is aminoacyl tRNA synthetase. Now, let us understand where does the proofreading take place. So proofreading occurs at step 2. Synthetase can activate wrong amino acid as well in some cases, but it will never allow that wrongly activated amino acid to attach with a tRNA. So proofreading occurs at step 2. Synthetase will not attach aminoacyl AMP to the cognate tRNA. Instead, the tRNA gets hydrolyzed from the active site if the match is incorrect. Now, this proofreading can be done by two different ways. There are actually two mechanisms by which this proofreading can be explained. The first mechanism is known as kinetic mechanism, and the second one is known as double sieve mechanism for error correction. In this part of our video, we will concentrate on kinetic mechanism of error correction. In the next video, we will discuss about double sieve mechanism. Let us move forward. Kinetic theory of error correction. The name itself suggests that it involves the affinity for binding and dissociation. So we are talking about the energies required for binding and dissociation. Therefore, this particular mechanism has been named as kinetic theory of error correction. The cognate tRNA, that means the correct tRNA, will have a greater intrinsic affinity for the binding site of the enzyme. So, they are bound more rapidly, but they dissociate more slowly. Once the correct tRNA has been bound, the enzyme scrutinizes the tRNA that has been bound. If the correct tRNA is present, the binding is stabilized by a conformational change at the active site of the enzyme. This allows amino acylation to occur rapidly. Now let's see what happens when a wrong tRNA has been bound by the enzyme or a wrong amino acid has been activated. If the wrong tRNA is present, the conformational change does not occur. As a result, the reaction proceeds much slowly. This increases the chance that the tRNA will dissociate from the enzyme's active site before it is charged. This type of control is known as kinetic proofreading. So, let us revisit this point. It says, supposing the wrong tRNA is present at the active site of the enzyme. Or you can say the enzyme has activated a wrong amino acid in step 1 and the tRNA which is there at the tRNA binding site is the correct tRNA for the enzyme. So match is incorrect. Okay. The conformational change will not occur in this case, which will not stabilize the tRNA acylation with the amino acid. Therefore, this theory is known as kinetic proofreading mechanism. Now, one more thing is also needed to be considered in this respect. The specificity of the amino acid varies in the synthetases. That means synthetases vary in their specificity for the amino acid. If similar looking amino acids are there, it is possible that synthetase can bind such an amino acid and may wrongly activate it. In the other case, a synthetase can be highly specific for the initial binding itself. It might bind the correct amino acid only, none of the other amino acid. But in other cases, some synthetases may bind to even closely related amino acids that are similar looking to its proper substrate. Although a wrong or similar looking amino acid can sometimes be converted into amino acid AMP, that means the adenylated form, but an incorrectly activated amino acid will not form a stable complex, that is the aminoacyl tRNA. Therefore, the tRNA will lose or will not form a stable complex. Therefore, it gets hydrolyzed from the active site. So actually, the summary of this theory is, if you have a correct amino acid, correct tRNA match, the energy required for binding is very less. Therefore, the affinity is more. And it will create the correct conformational change leading to aminoacylation or the charging of the tRNA. Whereas, if the tRNA or amino acid is not correct, the match is incorrect, then the binding energies are very high. Therefore, the binding or stabilization will not take place and dissociation will be faster. Let us understand this with much more clarity. The case 1 represents what happens when the correct amino acid and tRNA is there at the active site. The cognate tRNA binds rapidly, dissociates slowly. Cognate tRNA will induce the required conformational change at the active site, leading to charging. In case 2, supposing non-cognate tRNA binds, then this binding will be very slow and dissociation will be fast. Non-cognate tRNA will not induce the required conformational change, and therefore the active site structural change is not there, and therefore no acylation or very slow acylation, leading to dissociation of the tRNA from the enzyme's active site itself. I hope this is now very clear to you. tRNA synthetases don't just match molecules, they verify them and ensure only correct amino acid is selected and joined to the correct tRNA. This proofreading system is very essential for maintaining the accuracy of cellular protein synthesis. In this video, we have covered the kinetic theory of error correction. In the next video, we will discuss about the double sieve mechanism for error correction. If you like this video, please do not forget to share and subscribe. Thank you for watching.